Hello. Let's talk about how we store glucose in our body in form of glycogen. Before we start, make sure you know why we store glucose and why we withdraw it. Our purpose today is to understand first the process of adding glucosyl unit to the existing glycogen, and second, how to start the formation of glycogen. This is the structure of glycogen. In glycogenesis, we add glucose to this existing structure. An anabolic process that requires energy, we can describe it in 3 steps. These are the enzymes involved in glycogenesis. We have glucokinase or hexokinase 4, and then followed by phosphoglucomutase, and then we have UDP glucose pyrophosphorylase. This one is glycogenin, and then we have glycogen synthase, and finally we have glycogen branching enzyme. Recall your understanding on the class of enzymes. For instance, in this case, glucokinase. The function of glucokinase is to transfer phosphate to glucose. So this is glucose phosphorylation. And then we have phosphoglucomutase. The function is to transfer the position of phosphate from C6, carbon 6, to carbon 1, thus changing the structure. So what do you call that class? We have here UDP glucose pyrophosphorylase. The function is to activate G1P. This one is glycogen synthase. The function is to elongate the linear by forming alpha-1,4 linkage. Remember what is alpha-1,4? And finally, we have glycogen branching enzyme or amylo alpha-1,4 alpha-1,6 glucotransferase. The function of this enzyme is to help form an alpha-1,6 glycosidic bond. Okay, next we are going to take a look at the process briefly. To add glucose to the existing glycogen, our body activates the glucose to form UDP glucose. So this is an active form. You can see the glucose and the structure of UDP. So this is the precursor of glycogen. And this activated glucosyl unit of UDP glucose is then transferred to the hydroxy group at the C4 of the terminal residue of glycogen to form an alpha-1,4 glycosidic linkage. Mula-mula kita kena kenal pasti di mana dia akan terikat, di mana dia akan terikat. So therefore, we need a primer as a base to a glycogen structure. We need a base for it. So what is the base? So this is a protein structure called glycogenin. It functions as an acceptor of the first glucose molecule. So this is also actually an enzyme. We call it glycosyl transferase enzymes that will polymerize the first about 10 glucose molecules. After that, other enzyme will take over. Bermaksud glycosyl transferase ni ataupun glycogenin ni will help to polymerize, membentuk rantaian glucose. So roughly about 10 glucose molecules, dia akan buat linkage of that alpha-1,4 glycosidic linkage tu kan. Sebanyak 10 glucose molecules, lepas tu enzim lain will take over. Okay, these 10 units of glucose chain is attached to a tyrosine residue. Remember, I showed enzyme protein structure that contains sequence of amino acids. At that time, I showed you guys salivary amylase. Okay, as for this one, it has tyrosine at the position of 194 from all those 349 residue protein. So that's the position. So position tu lah di mana the first glucose will attach to. Sebab apa? Because of that tyrosine has hydroxyl group. So dia senang nak attach to that hydroxyl group. So now that we have 10 units of glucose that is attached to glycogenin, so what's next? Another enzyme, glycogen synthase, will take over the process. So glycogen synthase will form an alpha-1,4 bonds. So its action will result in a linear elongation of existing glycogen branches. These actually will help to chain up to 10 or more units. Some paper or some research mentioned that it elongate up to 10 to 13, some say 7. So we take the average line, so 10 or more units. After glycogen synthase built that glycogen chain of 10 or more glucose residues, another enzyme, amylo alpha-1,4 alpha-1,6 glucotransferase or branching enzyme to cut a terminal segment of at least 6 glucose molecules and insert it with an alpha-1,6 glycosidic bond on neighboring chain. So this part is known as branching reaction. Look at this structure. That round gray bits represent glucose residues linked alpha-1,4. The green one is glucose residue linked alpha-1,6. Itu ialah branching linkage tadi tu kan. So you can also see here reducing end attached to glycogenin yang warna merah tu. And then we also have that dark gray round bits, the non-reducing ends. Okay, this structure represents the idea of branching reaction. So at least 6 glucose, after 6 glucose at least, and then ada insertion with alpha-1,6 glycosidic bond. So dia akan branch. Selepas sekurang-kurangnya 6 glucose molecules akan ada branching. When we talk about regulation, of course, glycogenesis is also being regulated by the activity of enzyme. So enzyme di sini ialah glycogen synthase. So glycogen synthase, it exists in two form. Whether it is dephosphorylated, meaning dia aktif, dan juga in form of phosphorylated. So this phosphorylated means dia less active or inactive. Inactivation of glucose synthase is produced by a cascade of reactions, meaning many reactions involved. It is initiated by hormones similar to the activation of dia punya counter enzyme dia, which is glycogen phosphorylase. Okay, first, adrenaline and glucagon will bind to specific receptors on the membrane of muscle or liver cells. Kalau adrenaline akan bind kepada muscle lah, kalau glucagon akan bind pada liver cell. So what happened? This will cause activation of adenylate cyclase. Adenylate cyclase ni will catalyze the formation of cyclic AMP ataupun CAMP from ATP. Apa berlaku selepas itu? So, cyclic AMP ni, it will activate the protein kinase A. Remember protein kinase A? So, protein kinase A will phosphorylate a glycogen synthase. Bermakna, glycogen synthase is phosphorylated, so dia akan jadi inactivate. There's another enzyme called glycogen synthase kinase 3. Glycogen synthase kinase kinase 3 ataupun GSK3. So this kinase also can be activated by calcium ion, and it will also phosphorylate glycogen synthase. Kalau glycogen synthase is phosphorylated, maksudnya dia akan menjadi inactive. Another one is the role of glucokinase. So this enzyme is the first step of glucose phosphorylation to form G6P, especially when we have a meal rich in carbohydrate. So there'll be a lot of G6P created or produced. So G6P ni, it is allosterically activates the phosphorylated form of glucose synthase, so making it active semula. How are we going to use this information to explain the mini PRBL?